So the next group, or the next part of measurements, can involve what is known as the replacement series. The objectives or the research questions is that what is the competitive ability of plant species in a mixture? In other words, we want to know the competitive abilities of both the weed as well as the crop. So what is the competitive ability of weeds and crops, so that we get that information. Once we get that information, we then utilize it in weed management. Now, as far as the methods are concerned, you are going to have species proportions which are varied. So you've got the weed proportion and the crop proportions which are varied. But the species total is maintained. So the total number of weeds and crops should be the same in the end. So in a mixture of involving A species and B species, you can vary A from 0 to any other density, which could be X. Whilst you are increasing the density of A, you should be decreasing the density of B from X to 0, or just vice versa. I will show you the actual treatments. This is the example of plant mixtures in a replacement series. We call it the binary structure. If you are having maize and rapoco grass, for example, we can increase maize from 0 up to 10. So 0, 2, 4, 5, 6, 8, and 10. In that mixture, if maize is 0, it means we are having 10 plants of rapoco, and we can decrease them up to 0. So in the mixture where we have 2 plants of maize, there should be 8 rapoco, 4 maize, 6 rapoco, 5 where they are equal. It's an important treatment. Then you can have 6 maize, 4 rapoco, 8 maize, 2 rapoco, then 10 maize only, and 0 rapoco. So that is the binary structure of treatments. Sometimes you can also go up to ternary structure, but this is it may become complicated, So in the binary structure, in the ternary structure, you have now two weeds, rapoco grass and the Mexican clover. And the total may be 15 maize. You may have 15 rapoco, 15 Mexican clover, and you can have those various proportions. But somewhere you have a treatment where you have 5, all equally represented these plants. But we are not going to worry about the ternary structure, although it's much more realistic, but it can be very complicated as well. So we'll focus on the binary structure, maize and rapoco grass. The measurements which we are interested is the dry matter, biomass. How we are going to evaluate our information or dry matter, we will use number one, replacement diagrams. I will talk about replacement diagrams. Then we will also use competitive indices. Remember, there are more than 50 indices, so we just select a few, such as the following: A, relative yield total, relative crowding coefficient, and aggressivity. So if we focus on the replacement series diagrams, now on the x-axis, on the bottom, where you have got a mixture of A and B, you can have A increasing from 0 up to 1, it could be to 100. B, 1, it can start from 100 or 1, decreasing to 0. Then on the y-axis, you have got your dry matter. Now let us start with the A on that diagram. As you increase it from 0 to 1, A will increase, you get that slope A. If you do the same with the B, you start with the 1 or 100%, you decrease it up to 0, you get that slope going down. So you have got the slopes A and B. The total will be A plus B, that is the horizontal line above. Now, if you do an experiment with these treatments, where you have maize and rapoco grass with these proportions, now if you get such a diagram after drawing it and everything looks the same, it means A and B species have got equal competitive abilities. But if there are differences, you will get A being curved downwards sometimes and B being curved upwards. So A has got what is known as a concave shape, B a convex shape. If I go back to the first diagram, both A and B have got linear shapes, straight lines. It means both A and B have got equal competitive abilities. But if you have curves, if A is concave, it means it is less competitive. B is convex, it means it's more competitive. That's what it means. So you just look at the shape of those lines representing your species. In that scenario, B is now concave, A convex, which means B is now less competitive, or B or A is now more competitive. It depends on the sort of outcome which you are likely to get. Now, in this scenario, everything is concave. It means everything is being affected. We call this an antagonistic relationship. A and B are antagonistic to each other. They are equally affected in the system. So you may get a system where both the weeds and the crops are suffering. And that one where everything is convex, it means the system, the weed species are benefiting from that relationship. This is a typical example of symbiosis. And of course, I mean, with weeds, we rarely get this kind of relationship. If you may want to look at a paper, a replacement series evaluation of competition between rice oryza sativa and rice barnyard grass Echinoclea oryzicola. Now, in this study, they wanted to determine which rice cultivar was less affected by the weeds, or which cultivar was tolerant to the weeds. So these are the results, where we have a Shen, which is the rice, and the Delaman on the other hand is also the rice, competing with this wheat. Now, if we look at the curves, where you have that squared dot, I think it's representing the wheat, and the triangle is representing the variety. Now, you have to look at the shapes of the wheat and the crop. Let's focus on the first diagram. Now, if you look at the dotted straight lines, we can tell the shape of those curves. Now, Hashem looks convex, concave, and the weed maybe is convex. So the weed may appear to be more competitive in the first diagram, and then the second diagram, probably the other man is much more competitive than the wheat. You can always check this. Here we have got relative competitiveness of irrigated rice cultivars versus that wheat. Ashinomene denticulata. It's a wheat, I don't know it, but it was used by some scientists in an experiment. So this is what they got. If you look at some of these curves, you find that there's a lot of antagonism where you have concaves. So this is how they would have interpreted the information. Maybe if you have a chance of reading the paper, you may get to know what was happening. Again, a lot of antagonism, concaves there. Competitive ability of bean cultivars with the hairy beggar ticks. This is Biden's pilosa. Competition with bean cultivars, where you want to find the cultivar which is tolerant to this wheat. Again, a lot of concaves there, antagonism. Another paper, competition of soybean with blackjack, Biden's pilosa and pigweed, Amaranthus hybridus, done by Chivinge and Schwepenhauser. This was done here in Zimbabwe. They found that pigweed was more competitive than blackjack. Rupende also found that Rapopo grass was more competitive than Mexican clover using this approach of replacement series diagrams. So this is the summary of the outcomes, where diagram number 1 represents equal competition between two species. Number 2, where A is more competitive than B. Number 3, B is now more competitive than A. Diagram 4, everything is being affected. There's no dominant plant. Then number 5, everything is benefiting. So number 4 is antagonism. Number 5, where everything is benefiting, is symbiosis. So whenever such an experiment is done, either of the 5, any one of the 5 outcomes is a possibility. You can produce all the 5. It should be one of those outcomes. Here is these things can become complicated. You can see Chivinge and Mudzana 1989, where you have soybean in blue and acanthospermum in red. Of course, here it looks like soybean was more competitive than soybeans. I hope you agree with me if you look at the approximate curves from those diagrams. They may not be as smooth as some of the ones which we were seeing. So what are the parameters apart from replacement series diagrams? We can use relative yield of the crop, RYC. Relative yield of the crop is equal to yield of the crop grown in a mixture divided by the yield of the crop in a pure state. We also have relative yield of the weed, which is yield of the weed grown in a mixture divided by the yield of the weed in a pure state. These are some of the parameters which we can use. We will see how they can be used. Then relative yield of the crop plus relative yield of the wheat can give us what is known as the relative yield total. If I can just go to these diagrams where we say A plus B, that is what is known as the relative yield total. And on the y-axis, we have got either relative yield of the weed or a relative yield of the crop on both. This is on a single diagram. So how do we use relative yield total values? If it is 1, it means there is no competition, there is equal competition. Greater than 1, weed and crop are antagonistic. Less than 1, less than 1, it means weed and crop are antagonistic. More than 1, it means there is synergism or symbiosis. That is, if you calculate yield, relative yield totals, there's also what we call relative crowding coefficient. Let's say we have got maize and rapoko in a mixture. Relative crowding coefficient, in short, we call it RCC. If we want to find the relative coefficient, relative crowding coefficient of maize, we can get it using that formula. Maize yield in a mixture divided by rapoko yield in a mixture. We put that in brackets, divided by what is in the brackets on the right, which is maize yield in a pure state, divided by rapoko yield in a pure state. I think we can do the same for rapoko. If you want to find the RCC for rapoko, this time around it will be rapoko yield in a mixture divided by maize yield in the mixture. Then everything is going to be divided by what is on the right of this equation. Rapoko yield in a pure state divided by maize yield in a pure state. If you solve this equation, you get RCC rapoko. Now for these RCC values, relative crowding coefficient, that is what we call RCC. If it is 1, it means maize and rapoko have equal competitive abilities. Greater than 1, plant species in a mixture is more competitive. It could be maize or rapoko depending on the equation which we have used. If it is less than 1, it means plant species in a mixture is less competitive. It could be maize or rapoko, also depending on the equation which we have used. The other parameter is known as aggressivity, AGR. Now AGR for maize is 0.5, or in brackets, relative yield of maize minus relative yield of Rapoko. AGR of Rapoko, it's 0.5, or in brackets, relative yield of Rapoko minus relative yield of maize. Now let us look at the meaning of the values which you will get. If it is 0, it means plants in the mixture have equal competitive ability. If you get a 0 after using any one of the above equations. But the value could be positive or negative. If you have a high AGR, it means you have a strong competitor. If it is negative, it means you have got a weak competitor. What is the disadvantage of this method? The study is done at one density and cannot be extrapolated to other densities. That is its limitation. Otherwise, it can be useful. But if you want a more useful experiment where you can increase the densities, because the replacement series is limited, you can do what is known as the addition series. Don't confuse it with the first experiment, which is the addition experiment. The addition series, it can be complete, factorial, or a response surface. Or if you want, this is a hybrid between the addition additive experiment and the replacement series experiment. So the replacement series can be improved by including many densities in one experiment. The response surface diagram is used to interpret the results. The disadvantage is that this is likely to become a very big experiment. You may consider to read the Swanton et al. 2015. Now, what sort of treatments can we get? Let's say we were combining Rapoko grass and maize. We can work with various proportions. We can vary Rapoko grass from 100, we reduce it to 0. And the maize, we increase 0 to 100. And we can keep everything at 40 density. We do the same at 60 density, then at 160. In other words, we are having a proportion times density experiment. 5 proportions and 3 densities. It's a 3 times 4 factorial. If you wanted to find the design. In order to understand some of the outcomes of this experiment or the results, probably let us look at maize, or it could be a wheat. If you increase the density of maize, you are increasing yield. And there's some way where we can maximize yield by varying the plant population or density. And this is the equation which we can get: y equals to density divided by BO plus BC times the density, or in brackets. BC is the slope. BO is the intercept, and D is the plant density of the crop in the monoculture. Now, if you wanted to see what can happen to an individual plant, this is the graph which we get when we harvest everything together. Total effect of all the plants, total yield of all the plants. But if we're looking at the response of individual plants within the mixture, as you increase density, the contribution of each plant becomes decreased because of competition. Where you have few plants, each plant is contributing a lot. That's why we are having that curve. But if we plot an intercept, that is, if you divide the individual weight into 1 as the x-axis over density, you can get a straight line. So we can make that curve a straight line by taking the individual weight and dividing into 1, then we plot. We get things like that, where we have now BO and BC. BC is the slope, BO the intercept. So if we consider the reciprocal per plant weight, we get 1 over W, which is equal to D over Y equals to BO plus BCD. We are going to use these concepts later. Here we've got 2 students who actually did an experiment varying the density of Tithonia rotundifolia and Elucina indica in maize. First season, Munyaradzigata did an experiment which was repeated by Tavu Nyakurai in the second season. So this is what they were getting. If you increase density, you get that curve. But if you here, all of the Tithonia plants are being used. In this one, we are now using 1 Tithonia plant. You can see the curve, the curve has actually changed. And by plotting the reciprocal of Tithonia biomass, a straight line was produced. And that could be the equation for Tithonia biomass. The reciprocal of Tithonia biomass becomes a straight line, BO plus BD. Now we come to the addition series experiments. Now in a monoculture, which is this one, you can get a similar equation where represented by the crop, which is 1 over WC is equal to BO plus BC multiplied by DC. That is where you don't have the widths. Now, if you have got widths, the equation changes. For the crop, you now have the reciprocal 1 over WC, weight of the crop, equals to BCO plus BCC DC plus BCW DW. Those small letters are indicating they are giving us the labels for the crop and the labels for the wheat. That is the mixture. Now we also have the wheat, where we have the reciprocal of the WW equals to BWO plus BWW DW plus BWC DC in a mixture. Now those values, we can use them to calculate intraspecific competition, which is BCC and BWW, and also inter-specific competition, which is not which I can probably highlight from this. If it is inter-specific competition where we have two species, it will be BCW from the second equation. In the third equation, it will be WC. I hope you are seeing those small letters. That will give us inter-specific competition. So the advantage of the addition series experiment is that you can determine inter-specific competition as well as intra. You can determine intra-specific competition as well as inter-specific competition. You can also measure the competitive ability of the crop by dividing those indices. BCC over BCW, and the competitive ability of the wheat, BWW over BWC. And another issue, if you are an ecologist, you can also calculate what is known as niche differentiation index, as shown by that equation. So this is actually been done by Radovich in the methods to study interaction among crops and weeds. these are some of the treatments which can be used. The proportions, as I have indicated, this can become a very complicated experiment. You may need this kind of a graph, which is three-dimensional graph, where you look at the surface, where those red or brown squares, if you wanted to find out what is happening to your maize dry matter and the combinations of maize and rapopo grass. There's also a paper on weed crop competition experimental designs and models for data analysis, which you may want to have a look. And this was extracted from a typical paper where you see those reciprocal equations for different plant species and the graphs. And of course, interpreting the results could be a challenge, but after a careful look at the papers, you will begin to understand the meaning of those equations. Experimental methods for crop weed competition studies, if you are interested. This is a wheat and wild oats using these complicated designs. Response surface designs for investigating interspecific competition, another example. This was done in Nigeria, competition between Imperata cylindrica, a wheat, and maize in the forest savanna transition zone of Nigeria. Or another paper. Anyway, this was about the addition series. The other experiment which can be done is the dialer experiments. The research question is, what is the competitive ability of two plant species? That is, if you are interested in two plants. You can have such kind of treatments where you have two species A and B, which are grown in pure stands, as well as mixtures. For example, we can have A, then AA, and AB. You also have B, then BB as treatments. And you can easily measure intra-specific competition by comparing A versus AA, B versus BB. Inter-specific competition, you can compare rather, AB versus AA or AB versus BB. So that was that, very simple and straightforward. Then the neighborhood experiments, this can be very important. You want to measure the effect of weed position on crop yield. If you take a given field, is it important just to weed in row weeds? And we leave the between row weeds. I'm assuming that in row weeds are more competitive than between row weeds. Now, if you want to evaluate the method, you can have effect of weeds being assessed on crop which is the target species. The weeds are planted at various positions from the crop. For example, 0, 15, 20, 30, 35, and 45 centimeters. So you have got a crop row, and your treatment can be like this. 0, you have got weeds within the crop row. And you can have 15 centimeters. Weeds which are away for 15 centimeters. Then you can have weeds at 30, 35, and 45. And you determine what can happen to your crop. Now, when you vary the distance from the crop row, you tend to increase the yield, which means that the weeds which are within the row are probably doing more harm. There's a paper which was done by Nambagudzibwa. It's in, yeah. You may want to have a look at it. It was done for 2 seasons, and he was varying the weeds. At 0, you have got weeds within the crop. Then he had other weeds at 20 centimeters and 40 centimeters. In the first season, because of probably rainfall, actually where you seem to have more yield, it's where you have the weeds in the row. But if you look at this data, it wasn't significant. But in the second season, where we had significance, weeds in the row are depressing yields. And as we move away from the crop row, yield seems to be increasing. And this was actually done at the Henderson Research Station in Mazoe for 2 seasons. Again, this is another paper also looking at various distances. You may have a look at it if you are interested. Then we move to the nailed design, which is really more to do with forestry. If you are interested in forestry, this can be used in tree density plots. You can test multiple tree densities in a single plot. You have what are known as circular plots containing concentric rings, and regression techniques are used to analyze Nelda will studies. So this is a typical example of a plot, and those dots represent plant positions. Towards the center, it's more dense. As you go outwards, it's less dense, as shown here. More dense inside. As you go out, it's less dense. So probably yields decrease from the center, then they increase as you go outwards. This is a typical example of a plot. This is the actual plot under field conditions. Usually it's used in forestry, but sometimes under some situations it can be applied to weeds. Again, an example, towards the center it's quite dense. As you go outwards it's less dense. That is a photograph of the actual plot on the right. Application of nailed wheel experimental design in forestry research. If you are interested, you could have a look at that. Insights from full rotation nailed spacing trials with eucalyptus in Sao Paulo, Brazil. Another paper. Now I'm going to summarize what we talked about in these two lectures. Lectures on weed density. We defined the following weed crop interactions. By now probably we should have an idea of what is competition, adenopathy, interference, and parasitism. The other question which we addressed was that what do weeds or what do plants compete for? We also defined intra-specific competition and the inter-specific competition. Of course, we now know weeds can compete for light, carbon dioxide, nutrients, and water. And we tried to measure the effects of weeds on crops. So we have just discussed or gone through the experimental methods which are used to study the effects of weed density in crops. We started with additive designs, which are, I think, more realistic and more informative. Relative replacement series designs. Anyway, for the additive designs, we are interested in determining the weed control threshold. We are also interested in the competitive indices of the wheat, maximum yield loss, and the slope which we can attach to the wheat. Then replacement series designs, we want to find the competitive abilities of both the weeds and the crops. Now, addition series or response surface designs. This is a hybrid of number 1 and 2. We want to do the replacement series at different densities. And there are various ways of interpreting the results, some of the things which could be complicated. But you can calculate, you can estimate intra-specific competition and inter-specific competition. The diagonal designs where you are studying just individual plant species, maybe for academic purposes. These may not be useful for agronomic studies. Neighborhood experiments, if you want to study the effects of weeds relative to the position of crops. Maybe you want to target weeds which are more competitive, like the in-row weeds, which you may have to remove first, or you do band application of herbicides. Then the nailed experiments where you use the wheel-like structure plots to start the competition in the forestry. Indices of plant competition which we looked at: aggressivity, proposed by McGill-Christ in 1965, relative crowding coefficient, proposed by DeWitt 1960, Cousins and O'Neill 1993, then relative yield total, then also replacement series, substitutive experiments. This is the alternative name. What I just want to indicate is that replacement series experiments are also commonly used in intercropping studies, where you can intercrop maize and cowpea, maize and groundnut, but it's another matter. So this is it, I think, for now.
Thank you very much.
Right. So we've gone through crop with crop interactions. It's not yet 12, isn't it? Is it 12 where you are?
It's almost.
It's almost. Yeah, I think we've covered quite a lot. And there's also another portion which is not much, which we need to cover as well, under crop yield and under crop weed interactions. Remember, we need to know the weed density which justifies weed control. We also need to know the competitive abilities of crops and weeds. But we also need to know the time when it is appropriate to remove weeds. That we will need to answer as well, or we need to look at it as well. Maybe we may have a look at it later or now. What do you suggest? If we do it now, we finish about competition. It's not much. But if you want to finish exactly at 12, it doesn't matter. We can finish now. Are you saturated now?
Yeah.
I know Totoso time.
Thank you.
What I will do, I'm going to send you notes immediately. Then I'll also send you these video lectures. So together with some PowerPoint notes, so that you can use both. You can read the notes read other papers. You can also revisit the the lectures.
Thank you so much, Doctor.
Tochiclosa zvede. Any questions?
Yeah, none from me.
Thank you very much, Dr. Nyangwa.
Yeah, apa I think tine one question. You will see it after 3 weeks, I think.
Tinotera toka. Tinoiona.
Pane zvataita pane one question.
Takaimirira.
Ndikakupai manotsi, ndichakupaiwo assignment yamunozoita inoita follow up manotsi iwaya.
Okay, thank you.
The assignment inokubatsirai when you are in the exam, how to address some of the issues. Saka it will be a typical exam question for such kind of work. But it's not the question which you will find later. Okay.
Yeah.
All right then.
Thank you, Doc.
Tozoona narini Monday.
Yeah, I think Monday, tomorrow you're going to the fiscal lecture.
Mangwana mune chii?
Fiscal lecture, Dr. Garavaiti, tine practical, fasapanga so.
Hai, ndoda kumbosvika kushow. Ingangopera ndisati ndasvika kuka. Ndimboona kuti tiri papi in terms yeagriculture nezvimwe zvinhu.
Aiwa, ndizvo zvosvo.
Okay, thanks very much for your cooperation.
You're welcome to.
Thank you.
Thank you, Bob. Maswera zvakanaka.
Aiwa, pakanaka.